So good evening, everyone. Sorry, I didn't manage to post the material for yesterday's class yesterday. So I just posted it before the class. Go through it. And if you have any problems in, basically understanding it, do let me know. So the link for the second part, because in yesterday's class, we were talking about the antibody diversity, how that is caused, along with that HLA polymorphism. Briefly, we discussed about it. So I have taken that from Britannia Encyclopedia that I have posted a link for that. Go through it. Also, basic concepts of immunology also nicely explained. So together with that and immunological methods, the summary I made, okay, so should be sufficient for answering questions related to immunogenetics part. Okay. So where did we stop the class yesterday? I think yesterday we discussed about, I think we started rDNA technology, right? So genetic engineering and rDNA technology we started discussing about. So a brief overview I provided you with respect to genetic engineering, biotechnology, recombinant DNA technology. I briefly talked to you about the concepts related to GMOs, how they are helpful to us. This is a huge field, okay, where we have various different aspects that we may need to learn there. Okay, but most importantly, we notice that with respect to the biotechnology, few dimensions I feel are important. In case if they ask you, either with respect to the current scenario in India, how recombinant DNA technology or DNA technology in medicine is being used. Okay, let's focus on those components today. I'm going to talk to you a few specific things. Okay, this is to get a broader picture rather than getting deeper into one particular area, picking up few major advancements in the field of biotechnology and see how that is useful to us. Okay, and how that is helping us in the healthcare sector, most importantly, is what we are going to focus on. Let me pull up files for that. Okay. So gene therapy, stem cell therapies, and CRISPR-Cas9 technologies that we are going to learn as part of it. And also, I'm going to briefly talk to you about the ethical issues that are possible because in the last exam that we have seen that in the mains, they asked a few aspects of ethical problems as well. Okay, so we're also going to briefly look into those components. Okay, so let me share you this document. So how biotechnology or DNA technology is actually helping us in handling various different issues related to humans. So I think we have been talking about a lot of different basic ideas like DNA, genetic diseases, chromosomal abnormalities we were talking about. Plus, along with that, along with the inherited problems, we also have infectious diseases that bother us, correct? So in either of these cases, I think we need to have very good methods of diagnostics. And yesterday, as part of immunogenetics, we discussed about the immunological methods like ELISA method, or if you think about the Western blots, or blood analysis like complete blood picture, are looking for specific proteins, specific enzymes, maybe a sign of heart attack can be identified. PSA, we talked about an antigen that we normally look for prostate cancer. Like the detection of various diseases are becoming, more easier because of the advantages or advent of new technologies as part of DNA technology. So talk about them. Basically categorize the information into better diagnostics using DNA technology method. So these days we are talking about RT-PCR being one of the very well used and a common method of technology. So RT-PCR, right? So what is the general idea about RT-PCR? I'm going to briefly explain you this concept of RT-PCR as part of the better diagnostics. Okay, so understand them as theme like this. Better diagnostics, right? Either it is for infectious diseases or non-communicable diseases, those that are actually don't spread from person to person, like genetic diseases or cancers, those type of non-communicable diseases. For both of them, we are indeed working on bringing the better technologies. So recently, with respect to the COVID-19, we are talking about a diagnostic tool called RT-PCR. So what is this RT-PCR about? Excuse me. What is RT-PCR about and how does it work? Okay, I'm going to briefly show you an image on this. Yeah. So let me show you this image, which will make it a bit easier for you to follow the concept of RT-PCR. Have you heard about RT-PCR? So what do you know about RT-PCR? How does RT-PCR work? So what does RT-PCR stand for? Any one of you?
Reverse transcript.
Reverse transcriptase. What is reverse transcription?
Basically, maybe using the RNA of the virus or the pathogen and kind of creating a DNA or expressing the proteins or related to that RNA.
Okay. So basically detecting the RNA present in the microorganisms, and maybe microorganism is causing infection to us. So understanding that. Okay. So look at this image here. So RT-PCR is the, as you rightly said, reverse transcriptase or reverse transcription based PCR it is called. RT stand for reverse transcriptase. Okay. Reverse transcriptase. So RT in this stands for reverse transcription or reverse transcriptase. PCR stands for polymerase chain reaction. Okay. So first we'll understand polymerase chain reaction, PCR part of it, and then we'll go with the reverse transcription part of it. Because PCR is a standard method that can be used for not only something like COVID-19, because COVID-19 contains RNA as a genetic material. But even in case if they have DNA as a genetic material, PCR as it is can be used. RT is kind of an additional component to that. So what happens in polymerase chain reaction? I think I usually refer it to polymerase chain reaction as Xerox machine of DNA, right? Xerox machine of DNA, meaning if you put DNA into that and if you provide some components to it, DNA can get copied, like how documents get copied in the Xerox machines. Similarly, DNA copies can be made here. DNA copies can be made here in the PCR machine. So how does the PCR machine work? What is the principle behind it? The principle behind PCR machine, basically it is mimicking, so it is mimicking what happens during the DNA replication process. So it is mimicking DNA replication process in the body. Right, we discussed in unit 1.7. By any chance you remember how does DNA replication happen? So if you have DNA, normally double-stranded DNA double strands open up because of the role of an enzyme. And once it opens up, we have something called primers that bind to each sides, right? So primers are the short, fragments of nucleic acids, fragments of nucleotides, which are complementary to the template. So DNA template, which has to be amplified, it will open up and allowing the binding of short sequences called primers. These primers prime the process, initiate the process, because the enzyme which is responsible for performing DNA replication, which is called DNA polymerase, requires a short piece of DNA already attached to it so that that can be extended. So it's like this, like I think I might have told you this, if someone is building a wall, right? But the person who's building the wall has a problem that he needs first line to be fixed. Meaning you have to put few bricks, then he can join the bricks. So DNA polymerase is like that. So it requires few nucleotides to be added first. So in the form of primer, you add those sequences so that DNA polymerase can now read the template and make a copy of the template by adding nucleotides. Like that, it can extend the already partly synthesized DNA sequence, into a complete DNA strand. So like that, there are some requirements for DNA polymerase. If you satisfy those requirements, and you can take the DNA here, template will be the DNA that you want to copy, multiply. In case of COVID-19, we take a person's sample, swab test they normally do, right? So you've been seeing this. So they put a cotton swab into throat or into nose so that any sample that can contain COVID-19 is collected. Okay, so you collect the sample. There are two possibilities. One possibility is there may be COVID-19 in that person's body or may not be. So let's say there is COVID-19 virus growing in the patient's body. So when you collect, the COVID-19 sample should come out. And when you actually use it, so we are placing into a box, basically, a box in which you're you're trying to copy the replication, you're trying to copy the DNA. Okay, in this particular case, COVID-19 has an RNA. So that is why what do? They run reverse transcription first. Meaning simply reverse transcription refers to converting RNA into DNA. Because COVID-19 virus has RNA as a genetic material, and from RNA you derive DNA. Because complementary sequence to RNA can be created, right? Thereby making DNA. So by reading RNA sequence, DNA is made. That is the first part, reverse transcription part. So that is why we require this additional component attached to the PCR part. So where RNA is first converted into DNA. Otherwise, rest of the concept is same in bacteria, same in anything that is to do with DNA. Because now you derive DNA from RNA. That is the one extra thing you have to do with COVID-19 or any other RNA containing viruses. Once that happens, so your goal is to mimic the DNA replication process, thereby make copies of DNA so that you have enough DNA that you can quantify or enough DNA that you can see to tell that yes, this person is suffering from COVID-19 infection. Okay.
Sir, this reverse transcription. So, irrespective of the name, every time it means that RNA is converting into DNA.
Yes.
Okay.
Reverse to transcription. So transcription results in, basically transcription is a step, the first step, right, in gene expression. From DNA, you make mRNA. From mRNA, you create proteins. The first step is transcription, correct? So reverse to transcription, reverse to formation of mRNA. So instead of making mRNA from DNA, you are going other way around. Reverse to the transcription process. That is what it means. Okay. There is an actually enzyme called reverse transcriptase. RT or reverse transcription is done by enzyme RT or enzyme reverse transcriptase. The enzyme itself is called as reverse transcriptase. What do these people do in the laboratories? They collect the swab sample. To that they add RT first. RT will convert RNA into DNA. Then they put the DNA into this magic box called PCR. So what happens in this magic box? So you have to add a couple of things to it so that DNA replication process happens. So basically add whatever normally is present inside your body for allowing DNA replication process. So what are they? So look at this image. So what do we need for DNA replication to happen? We need DNA sample. Where is DNA sample coming from? The swab sample that normally take, which should contain the source of genetic material. For RT-PCR part, you run RT so that RNA first converted into DNA. That's how you obtain the DNA sample. Wherever you are unsure about what I'm talking about, do stop me there. Okay. So this is, you already explained about the DNA part. Correct? Okay. Then what else we have to add? Primers. I'm assuming that you remember the concept of DNA replication. So some of them I'm quickly explaining it. But if you lost the idea, do let me know. So primers is to start the process of replication. And then what do you add in the process of making DNA copies? Basic units, building blocks of DNA, which are nucleotides. Plus, we need an enzyme. Don't worry about the tag or anything. Forget about that. Polymerase is an enzyme. DNA polymerase we normally call. TAC is a commercial name of that enzyme. Polymerase, DNA polymerase. So DNA polymerase is beginning to add nucleotides where primers are bound. But how does it know what nucleotides to add? It looks at the template DNA. In this case, DNA, in case if the person is COVID positive, the COVID DNA is seen. So how does this reaction differentiate between human DNA and COVID DNA or COVID genetic material? Because you're collecting swab sample, right? It should also contain our cells. And also, in case if there is COVID infection, COVID virus will be there. Which one gets amplified by this machine? Because we have DNA of ours. I mean, even more than this, you may have so many different types of bacteria in your throat. Even they have DNA. And if you are going to multiply the DNA, whose DNA will be multiplied? Bacterial DNA, human DNA, only COVID DNA, or COVID derived DNA, because COVID doesn't have DNA. COVID derived DNA. What do you think?
I think everything gets multiplied. But
Everything gets
again, in the box, probably we'll use something like ELISA's principle.
So we use additional method to detect the presence of it. So we need both RT-PCR and ELISA.
Yeah, that's what I'm thinking.
Okay. Is there anything that adds specificity to the reaction? Anything that specifically allows the replication of only one type of DNA strand? So what is giving specificity here? Doesn't matter. You try whatever you want. Okay, that's all right. I'm going to explain that, but just want to know where you are.
Buffer.
Buffer. Okay. Buffer is like environment. Buffer only
Okay.
contains salts, all sorts of things. It's environment. Yes, Sunil, you're trying to say something?
Sir, the proteins that we have should be different from the protein of.
Okay. So what is the role of proteins here?
Sir, in the transcription, the mRNA converts it into
Yeah.
readable form. So our protein should be different from theirs. My just guessing here.
Okay.
At least from picture, maybe temperature.
Temperature is mainly to allow what should happen next. So in the image that you see here, so the temperature variation happens. That is why this is called thermal cycler or thermocycler they call, because this machine allows us to change the temperature. But change in temperature would only allow what happens at a given point of time. The specific point here that you need to know is primers. Primers that we use here, not random ones. The primers that we use here are specific to COVID-19. So what we are doing, we are selectively multiplying COVID-19 genetic material by using primers which only match to COVID-19 genetic material. If you want to, like let's say you want to make a copy of insulin gene, we were talking about taking insulin gene. Let's say you want to use PCR for making copies of insulin. What do? You take complementary sequence to the insulin gene. The primer should match to the target. So only the region to which primer binds, that region gets amplified. So DNA polymerase job is to add the nucleotide to wherever primers are bound. Where would primers bind? Primers will bind only when there is a complementarity. So we have, assuming that we have human DNA sample, bacterial DNA samples, viral DNA samples, all of them will be there, but only what we defined. So here, all of this is common to any amplification process. Enzyme is same, buffer is same, nucleotides are same, but what is different? The primer is different. So you can use same RT-PCR test for the detection of dengue, Zika, chikungunya, HIV. Okay, but what will be different? The primer sequence will be different. If you're testing for HIV, primer should match to HIV. If you're testing for TB, primer should match to TB. If you're testing for any other known disease, primers are specific to those pathogens. So yes, we actually selectively amplifying it. We are not amplifying everything, or we are not trying to filter it later, because that's use wastage of resources here. So instead, what do? Primers are very, very specific. So primers are the defining factors. So imagine if you use, let's say, COVID-19 primer, but there is no COVID-19 in the patient's body. Will that amplify? Will there be anything for primers to bind? No. So similarly, you choose, test is defined based on the primers. Rest of the things can be same. In the laboratories, what we normally do, we have all these components common. Primers, we make orders. Companies are there, we make orders specific to matching particular sequence. We make primers specific to COVID-19. That is what companies are doing. Companies sell you a package where primers specific to COVID-19 are given in the package. Rest of the things we can use them, even make them in the laboratory. Okay, so specificity component is coming from where? It is coming from the primers part. That is where we are filtering it. So you're not amplifying everything, you're only amplifying selectively COVID-19 genetic material. As PCR is meant for DNA, so we have to convert RNA into DNA first. Okay, right. Rest of the thing is more like the steps that you have to follow in DNA replication. So briefly, what happens here? So mimicking DNA replication. What happens in the body in the DNA replication? First of all, denaturation happens. DNA strands have to separate, unwind. DNA helix has to unwind, right? That is called denaturation. So first step is always denaturation. How do you achieve denaturation? You heat up the sample. You heat the sample to 95 degrees centigrade. As soon as temperature hits 95 degrees centigrade, DNA starts opening up, allowing the primers to bind and polymerase to add the nucleotides. So first step is denaturation. Denaturation means separation of DNA strands. First step is denaturation, separation of DNA strands. Look at how well they designed this. The scientists received Nobel Prize for designing this machine. Biotechnology, many things in biotechnology are becoming possible today because of PCR. So very, very important tool and one of the essential tools in biotechnology. So first step is denaturation. Okay, separation of DNA strands. Then what happens? You reduce the temperature to 50, 55, around like that, depending on primer length. 55 degrees or approximately 50, 55 degrees. What happens here? Primer binds to the template. So denatured template now is accessible to the primer. So primer will bind to the template. So that is called annealing. Annealing means binding, pairing. So second step is annealing. Binding of the primer to the template. So what has to happen next after primer binds? The next step should be polymerase adding nucleotides to the primer by looking at the template DNA sequence. Here template DNA sequence is the COVID DNA sequence or COVID genetic material sequence. So what does this do? So these primers get extended, annealed primers get extended by addition of nucleotides by the polymerase enzyme. So this is called extension. This is called extension. Denaturation, annealing, extension. These are three key steps that happen in the PCR, the PCR machine. So like that, the thermocycler is designed in such a way that temperature can be varied. You program it. We can actually program this machine. So first few minutes 95 degrees, second few minutes 55, next 72. So depending on the concentrations and the actual substances available, these reactions take place. Like that, you do multiple runs. So each end of each run, you will have two DNA strands are formed, right? From each strand, new strands are formed, two new strands. But that's not enough. Having this double the amount is not enough. What do? You actually multiply it 10 times, 30 times, 40 times. You will exponentially make millions of copies of DNA. How much time? 1.5 hours to 2 hours, depending on the size of the DNA. In very few minutes, maybe hours. That is why RT-PCR takes so much time, because they normally, I think, processing and preparation and extension requires about 3 to 4 hours. That is why RT-PCR is a slow method, time-taking method, because they have to go through all these steps, not once, multiple times to see enough DNA. But this is a basic idea about how PCR works. And why is it called polymerase chain reaction? Do you see the link here in the explanation part? Why is it called polymerase chain reaction? Excuse me, why is it called polymerase chain reaction?
Polymerase is used to amplify the DNA.
Correct. So polymerase is a key player. It is an enzyme. Very good. Next. Chain reaction.
DNA is duplicated.
DNA is duplicated. Okay, in what sense it is chain reaction? What
The
is it?
available DNA is again denaturated, annealed, and extended.
Very good. Chain reaction means where the product of previous reaction will act as substrate. Like, I don't know if you studied about this in nuclear reactions. Chain reaction happens in nuclear reactors. Neutrons that are produced in reaction would split more uraniums. Correct? So chain reaction is where, let's say, a product is formed. This product will now act in the next step. Because every time new products are formed, you have more exponential production of the next set of products happen. This is called chain reaction. Each step is connected to the previous reaction. So each reaction depends on the previous. Like that, in chain, they start producing exponential amounts of DNA. Polymerase is the enzyme, and it results in chain reaction because every time a new DNA products are formed, they will act like templates. That is, otherwise what happens? 2 will become 4, 4 will become 8, 8 will become 16. They become exponentially, this results in exponential amount of DNA. Because every time the product is made, it will become substrate for the next one. So first you have only one DNA strand, second you have 2 DNA strands, 4 DNA strands, then 16, like that. It will continue to amplify like that. That is called polymerase chain reaction. Okay, so this is considered to be very, very sensitive test for COVID-19. The COVID-19 scenario, we are able to detect it authentically because unless primer matches to the genetic material, no amplification will take place. It's a very, very accurate method. This is considered to be gold standard in detection of COVID-19. Unlike imagine with the rapid antibody, rapid antigen test that you hear about, where you look for antibodies in the body against COVID-19. So you collect the blood, take plasma and look for antibodies that our body might have made against COVID-19 infection. But these antibodies are time sensitive. So they only appear after a few weeks of the infection and they may stay or may not stay after a few months. So it is very difficult. And when you're checking for antibodies, it doesn't tell you whether someone is actually suffering from infection at the moment. Antibodies are already made. You maybe just had an infection a few weeks ago or you had infection last month. The active infection will not be known. So with the rapid antibodies, this is one big problem. So although rapid antibody tests are cheaper because they happen to be around 500 rupees or even less than that, but this RT-PCR, because of the requirements, all these steps and those machines, they initially costed us 4,500 rupees. But today, I think I remember in Hyderabad the last month when I got tested, 700, 800 rupees, 700, 800 rupees per test, where it was 4,500 rupees a few months ago. Because of the laborious nature of this process and the number of steps that you have to go through. Okay, right. So this is a very good diagnostic. Such diagnostic tests are coming into the picture now. ELISA had become very popular. National Institute of Virology had made ELISA for COVID-19 detection as well. It's a bit better than regular antibody testing. That is also antibody antigen testing, but better. Okay, so PCR is now becoming a common tool. India also recently, I think WHO recognized India's PCR test for TB detection as one of the most sensitive tests. So like that, India is also pioneering in this field. We are developing because there are so many biotech companies in India. We are able to produce and create better diagnostic methods. For TB it is famous, for HIV it is famous, now it is becoming famous for COVID-19. Okay, like that better diagnostic tests can be designed using DNA technology. All this is DNA technology, right? You realized how DNA replication works, created some pieces of components like primers and polymerases, nucleotides, and run the test in the laboratory. And you are very specific and accurate in determining whether you have so-and-so pathogen in the body. So like better diagnostic tests are being designed as part of DNA technology. Okay, right. Next, let's focus on the treatment options. Let's focus on these treatment options. So first I'll start with the stem cell therapies. Excuse me. So brief idea about stem cell therapy. We don't get into the details, but I'll tell you how stem cell therapy works. So what are stem cells first of all? What are they? So can someone give me a definition of stem cells?
The cells from which other cells get formed.
Cells from which other cells can be formed. So to be more specific, your idea is correct. To be more specific, here we talk about stem cells are the cells which can differentiate into, all types of cells. Okay, the keyword here is differentiate. Differentiate means becoming a cell, a new tissue. Okay, or so basically they transform into a new tissue. They differentiate into new cells. So stem cells are the cells which can differentiate into all types of cells. So they can form basically all types of tissues and even organs in the body. Correct? The cells which can differentiate into all types of cells and can form all types of tissues or organs in the body. Such cells are called stem cells. Any observation that you can make where you know stem cells are indeed present in our body? Any argument that you can give which tells us that indeed we have stem cells? Any observation that we discuss these type of observations in the classes?
So any injuries
Hmm.
recovered? I mean, any wound gets recovered within some days.
Okay, but that is probably not necessarily due to stem cells. Like for example, your skin cell, you got damage to skin cells. Skin cells replicate. Skin cells will give rise to new skin cells. It doesn't have to be a stem cell, right? Hair loss. If we have, cells that hair cells that multiply,
So
something more.
from when the embryo is in the womb, maybe at different stages, the organs are not formed,
Right.
but then they are formed.
Exactly. Very good. So that is the best observation that we can think, we can talk about here. Yes, when we are born out of fertilization process where we started life with a single zygote. Zygote became embryo. Embryo has multiples of cells, hundreds and thousands of cells. And embryo only had cells, no differentiation. Embryo, if you open the embryo and check it, it's basically collection of cells, group of cells, undifferentiated cells. But eventually embryo will give rise to fetus. This is where all sorts of tissues, body parts starting to appear. So meaning the cells that we had in the embryonic stages have the ability to give rise to various types of tissues. Heart gets formed. There is, I think, a great documentary on YouTube by National Geographic, how we are born. They actually show clearly that slowly various parts in the body gets formed. Your heart, your kidney, your spine, all parts eventually starting to appear because we look more or less like a fish initially in the mother's womb, and eventually we appear to become more like humans. So where are the cells coming from? The existing cells in the embryonic stages. So they are giving rise to these new tissues. So we definitely have those cells in our body. Otherwise, we won't grow, we won't become, we will not have these organs. What our parents give to us is basically their software. They're basically giving us their software. So we are adding the genetic material of the mother and the father in mother's egg cell where environment is there. But they're not giving us their organs, correct? They're only giving us instructions. With instructions, we are able to make whole organism because along with the instructions in the initial stages, the cells that we have in the embryos have ability to give rise to all sorts of tissues. Such cells are called stem cells. Okay, are you able to follow this? Is the concept clear to you? Okay. So there are the different terms. I don't want to get into the details, but just want to mention to you, depending on their ability, stem cells can be of different types because we don't have those stem cells. Do we have those stem cells today? Do we have any stem cells at all today? If you ask these questions, we actually have the stem cells which have some limited ability today. We don't have same stem cells that we had initially in the embryonic stages. Those stem cells are called totipotent stem cells. We had initially totipotent stem cells, meaning they have total potential. They have complete potential to give rise to an organism. They have complete potential to give rise to an organism. They can give rise to complete organism. They can give rise to complete organism. You call it totipotent stem cells. But over a period of time, the ability of the stem cells will go down. We will have what is called pluripotent stem cells. Pluripotent stem cells will eventually have pluripotent, multipotent. Like that, the ability of the stem cells go down, decreases. Ability of the stem cells decreases. So today, we probably don't have these stem cells in our body, but something that has limited ability. Like if you can use the word oligopotent, or we have ability to produce few types of cells today. The example, if I have to take here, is our bone marrow, stem cells in the bone marrow. You all know what is bone marrow?
No, sir.
Yes.
Bone marrow is a soft part in the bone, right? I'm not sure if you're vegetarians or non-vegetarians. If you're non-vegetarians, you probably know that soft portion. I'm a vegetarian, so I don't know. I only heard about it. Okay. They say that the soft portion or a nicely tasting portion in the bone is bone marrow part, the central part of the bone. Okay. This is where our blood cells are formed. RBCs, WBCs, platelets are formed within that bone marrow, within that central part of the bone. That region is called bone marrow. Okay, we have stem cells in that part. We have stem cells in that part, but they have ability to give rise to only few types of cells. Like if you know, bone marrow is a place where our RBC or WBC and the platelets are made here. Or RBC, WBC and platelets are made here.
So these stem cells can also help to cure cancer or chronic diseases,
Stem cells also help in.
like to cure the cancers or chronic diseases.
Yeah, I'll come to that. Okay, let me first give you the concept of stem cells, then I'll talk to you where can we use stem cells. Okay, so what I'm trying to tell you here, most of the stem cells that we need, we probably have them or had them in the embryonic stages, but as we pass on or we grow up, so the ability of the stem cells decreases from the stem cells which have ability to give rise to entire organism to stem cells which can only give rise to few types of them. Like in the bone marrow, we have RBC, WBC, platelets producing stem cells. Okay, like that. So we have several stem cells in the body, and even scientists have found out this is a great advancement that happened in the biotechnology part. Is some scientists from Japan, they have created induced pluripotent stem cells, meaning they actually created stem cells from normal cells. So this is again Nobel Prize winning research. So Japanese scientists, they have created induced pluripotent stem cells. So what have they done here? They took, like, say, normal differentiated cells. Like for example, I've been so far telling you that stem cells will give rise to various types of cells. Stem cells will give rise to various types of cells. Correct? But what they have done, they took the differentiated cells, like our skin cells, our muscle cells. They took those cells, they reprogrammed them. So what they have done, they took the differentiated cell, they took the differentiated cell and differentiated cell they converted into a stem cell. So this is through reprogramming process. What they have done here, they reprogrammed them. It's like, reprogrammed. It's like hypnotizing them. It's like you go to a skin cell and you say, you are a stem cell, you are a stem cell, you are a stem cell. It became stem cell. Will that happen like that? Okay, so basically they reprogrammed in a sense. They added these signals. They added the proteins and signals because normal cells and stem cells are in no way they are different from each other. They have same DNA. You take a stem cell, you take our normal cell, skin cell, they have same DNA. DNA doesn't change except for mutations. But what has changed? What signals are you sending? It's like this. Let's say in a family, two kids are born. One became engineer, another became a doctor. Right? Common theme in India, right? Either engineer or a doctor. So engineer thinks like an engineer. Doctor thinks are trained to become a doctor. So basically, they're trained to become something like that. Same way, stem cells are trained. Stem cells are actually given certain signals to make them act like stem cells. But DNA is same. Genetic material is same in both of them. In both the kids as well, genetic material has come from same parents. But the way they're trained, their environment has changed them to become something else. Similarly, the environment of the cell is important. These scientists have found out that environment. They gave those signals, and this cell, normal cell, started behaving like a stem cell. And this process is called simply a reprogramming process. Reprogram the normal cell to become a stem cell. And these stem cells are called induced pluripotent stem cells. Like that, scientists have created stem cells which we can use today. You heard about stem cell bank stories, stem cell banks. So those type of components you hear these days. Okay, even if you don't store stem cells, I think technology is coming up. I don't know how much available this technology is to us, but you can create stem cells from normal cells. This technology had been figured out by scientists. So now we move on to the applications part, uses. So this is a brief idea about stem cells. Where are we using them? Where are we using the stem cells? So look into the features and ability of the stem cell. Stem cells have ability to give rise to any types of tissues. The simplest thing, if someone burned their skin, can we regenerate the skin? Someone burned the skin. Okay, or someone is a acid victim. Nowadays we hear so many stories of acid victim, right? So what you do here, so regeneration of skin or skin grafting, meaning you actually take stem cells in the laboratory, grow skin tissue, and use this tissue to cover up the body that got burnt or damaged by acid. So skin grafting can be done. So the skin can be produced by stem cells. Hair, nowadays we are hearing about hair transplantation. Here is, of course, hair doesn't cause us too much of a physical problem, but more of a psychological problem. You agree with it? People are more worried that they don't have hair. It's a psychological problem, correct? So, but of course, psychological problems can lead to many other issues. So, stem cells can provide as a solution to this. Here, people can actually have stem cells that are used to grow new hair. So you can grow hair, you can grow teeth. You think about any tissue or organ, we should have stem cells that have ability to give rise to that. So in a very basic case, regenerating skin, regenerating hair, regenerating teeth, or producing teeth can be done with. More useful than that is bone marrow transplantation. Bone marrow transplantations. What happens in bone marrow transplantations? Let's say a person suffering from a rare genetic problems or some problem where his immune system cells, WBCs are not made, RBCs are not made. So you can collect bone marrow sample, they normally inject, put a needle into the bone marrow in the center of the bone, collect some of this bone marrow or this material, inject into another person. So the stem cells that you collect from here can be given to some other person, and stem cells, given that they have ability to give rise to new RBCs and WBCs, platelets, they can do that. So bone marrow transplantations are common and well established technologies. Since last at least 3 decades, bone marrow transplantations are in use. Okay, so this is also an application of stem cell technology.
So, the bone marrow or stem cells are directly taken from one person and given it to another, and they act on their own.
Stem cells from one, like basically bone marrow, if you're collecting, stem cells present in them are transferred to another person. So what happens, those stem cells can have, given they have ability to give rise to blood cells, they do it. But there's little problem here. If you're taking someone else's stem cells, again, compatibility becomes an issue. Yesterday we talked about MHC, right? So the cells that carry different MHC, there's no matching, there'll be a problem. So they would, I think if I'm not wrong, they might do some sort of matching has to be done here. And only for when it is compatible, found to be compatible, along with some immunosuppressive drugs, they give. So like that, stem cell bone marrow transplantations can help in regaining the missing WBCs, RBCs, etc. Okay, so you can create various tissues, human tissues. Human tissues. We are not there yet, but human organs can also be made. I think recently, in combination with 3D printing technology with stem cells, they're able to print something that looks like human heart. American scientists, I think, December 2020, they printed human heart. In those type of experiments, also they utilize stem cells to give rise to the tissues that look like human part. So human tissues and organs can be created. Okay, most importantly, we have what is called regenerative medicine. Regenerative medicine is becoming popular these days. So in future, I think hopefully you can create an organ. Let's say someone is suffering from kidney problem, kidney damage is happening in a person. In future, maybe we can order kidneys. You tell them these are my stem cells, okay? Can you please make a kidney for me? You may be able to order in future. I don't think in our lifetime we may be able to see it. Okay, so organs may come. Tissues are already in place. You can make tissues. I'll tell you in the next example how tissues are being used these days. But otherwise, regenerative medicine is slightly different concept. Is where I think scientists at Darwin Institute of Medical Sciences are working on it, where they are working on injecting the stem cells into human body. Let's say someone is having heart issues, heart problem, heart damage has happened. Heart is one such a tissue which doesn't replicate. If you get a heart, those cells in the heart, they don't replicate again. We get what we get. Okay, so if damage happens, that's it. That's the end of our life. Either you can surgically fix it or mechanically put parts into the heart, but you cannot repair the heart as such, or it doesn't get itself repaired. So what do we have to do? Scientists decided to inject stem cells into this patient's body whose heart is damaged. And interestingly, I was reading this in Science Reporter magazine once. So where they said stem cells, when injected, they find the damaged part of the body. So they are directed to the damaged part of the body themselves. They have some signals that actually take them to the damaged part. So it will go to heart and they start repairing heart. So through stem cell therapies, especially in the regenerative medicine or regenerative therapies, scientists are working on just injecting stem cells. They will fix the problem. Either it is heart problem or it is nervous system problem, neuronal issues, neuronal problems, like neuronal problems in the sense like let's say Alzheimer's, Parkinson's. For those type of diseases also, Alzheimer's, Parkinson's, etc. And there are a few more. Stephen Hawking died with ALS. So those type of rare problems can also be cured using stem cells. Okay, amyotrophic lateral sclerosis, ALS. Okay, one rare disorder that Stephen Hawking got. So those type of problems can be cured in future using stem cells. So great potential. I think once it came in the paper 3, in mains paper 3, stem cells are panacea for many human problems. Examples, they gave list of problems. Okay, like that. Stem cell technology is right now redefining the treatment methods. Okay, the last example I'd like to give you are drug testing. We can also use them to test drugs. Like for example, cancer. Let's say someone found out cancer drug. Okay, and you already tested on animals, but there is no guarantee that if it is showing positive result with animals, there is no guarantee that it will work on humans. So before trying on human volunteers and after trying on the animals, we have to use something that is more like humans, like human tissues. Take stem cells, grow liver tissue, grow kidney tissue. Test drugs on them. See if these drugs are killing those tissues, human tissues. Then toxic effects can be already figured out before trying on human volunteers. Otherwise, we sometimes hear that 3 of these people died, 4 of these people died when during the drug testing. So we can avoid even that. Okay, like that, stem cells can provide us new means of creating tissues, organs, and repairing these organs in our body through regenerative medicine, where they cannot themselves gets repaired, or testing the drugs. So cancer drug can be tested on tissues, and tissues derived from stem cells. Okay, right. And answering Shweta's question, Shweta, as I was asking, if I'm not wrong, can we treat cancers? Okay, so along with the next topic I'm going to talk to you, along with stem cells, in combination with gene therapy, we can also treat cancer problems. So what do they do here? You may want to make changes to the gene that is causing problem. Let's say someone got breast cancer because of BRCA1, particular gene called BRAC1 received the mutation. So what do? You actually change the mutation, switch the mutation through gene therapy, or replace the defective gene, mutant gene, with a normal copy of the gene. And what do you modify here? What is the role of stem cells here? Do this modification in the stem cells. Take stem cells, make changes in the stem cells, put it back into the patient's body. What happens? Patient will start to, produce the normal version of that gene, causing no cancer. So like that, cancers can be treated. And nowadays we are hearing it as part of immunotherapy that I'm going to discuss in the next part. Immunotherapy, gene therapy that we are using are currently experimented, and specifically called immunotherapy is being used for cancer treatments. More safer and more effective. I'll explain you how. Okay, but for now, just make a note of this. So this is about stem cells. Do you see how stem cells are being beneficial? If they ask, I think mostly so far I've seen questions being asked more generically. Talk about the role of DNA technology in treating human ailments. The questions are asked like that. Pick a few examples like this and write about it. You don't have to give details. What stem cells can do, what next we're going to talk about gene therapy can do, what GMOs can do, therapeutic products in genetically modified bacteria. So add diversity to your answer.
Animal artificial meat can also be one example.
Artificial meat can also be given as a handling food scarcity or ethical issues. Maybe you don't want to kill animals for human use. Then artificial meat or clean meat, lab meat they're talking about these days can also be definitely mentioned. Depending on the demand of the question. Clean meat, lab meat is to supplement for the foods that we are eating with more protein and with less killing of animals, we can do that.
So artificial meat is made to these stem cells.
Not necessarily stem cells. What do they do in artificial meat is they collect a tissue. So artificial meat or lab meat is called. So in them, they take tissue, animal tissue, let's say. They take animal tissue and basically in the laboratory conditions, what they perform is tissue culture. So they grow cells. It doesn't have to be stem cells for that because you don't have to grow various parts of the body, right? So you can take tissue from the animal cell, grow this in the laboratory conditions. So like plant tissue cultures, plants can be grown in the laboratory. Similarly, this is animal tissue culture. You take animal tissue and grow them in the laboratories. So bulk of the animal tissue can be obtained. Maybe you can design in such a way that more protein content is maybe present, less fat may be present in it. Like that you can redesign the meat. But as this meat belongs to animal, because it has a nature and features of animal meat, but it is grown in the laboratory, it is called a lab meat. So Singapore, I think, has introduced the lab meat recently, right? And America also has it. India wants to introduce it as well in a few years. So it is not necessarily an application of stem cells, but application of DNA technology. Okay. So next, let me briefly tell you about the role of gene therapy in dealing with this. If you talk about gene therapy, no, I don't think so, right? So what is gene therapy? So in genetic diseases, we learned that mutations in genes, mutations in some essential genes. So mutations in essential genes cause this problem. Correct? So because of the mutations, it results in defective genes. It results in defective genes. Can we replace those defective genes?
Yes.
So that is what the idea. In gene therapy, your goal is this. Your cell has a defective gene. You want to add a healthy copy of the gene in place of it. Either you remove the old one and replace it with this, or you add the functional copy of the gene. So whatever function is missing because of the mutation. Because after mutation in hemophilia, clotting factor, a particular clotting factor may not be formed, right? In the hemophilia example we talked about, either factor 8 or factor 9 is not formed. When factor 8 and factor 9 is not formed, that clotting process doesn't finish, doesn't take place. But what you actually add a gene which will produce now clotting factor. So you add a normal copy of the gene, clotting factor is made, the clotting factor will finish the reaction. Correct? So we can do something like that. So gene therapy is that. Gene therapy is where you are compensating for the missing function of the defective gene. Either you add a copy of the gene, or there are other ways to it. I don't want to complicate this unless you have some knowledge on this. So defective copy defective copy can be replaced, or a functional copy is placed into the cell so that you can revive the missing function, regain the missing function, is what is gene therapy. It is giving a healthy version of a defective gene to cure a problem, to cure something like a genetic disease. So how is it done?
Usually through screening and then maybe at the embryo or early stages of life.
So good. So at the early stages of the life, I think this is preferable to do. But what cells do you choose? Let's forget about embryos for now. What cells do you choose? Do you take somatic cells or germ cells? Which are preferable?
Germ cells.
Germ cells. So there are two types of gene therapies: germline gene therapy, somatic cell therapy. Germline means you target germline, germ cells, so gamete producing cells. Somatic cell therapy means you target somatic cells. So somatic cell therapy and germline therapy. We have two of them, two types of them. Okay, the few differences are listed here in the slide that you see. So in case of somatic cell therapy, you introduce genes either into bone marrow, blood cells, skin cells, any somatic cells, leaving out those gamete producing cells. That is what's called somatic cell therapy, where you're introducing genes into skin, blood, bone marrow, muscles, etc. Germline is introducing genes into eggs and sperms. But what is the difference? Why do you want to? Because germline, if you introduce into germline, so they get passed on to their later generations. Whereas if you introduce only into the somatic cells, they remain in that generation. So which is more preferable? Germline therapy or somatic cell therapy?
Germline.
Germline, okay. But in
I think
you, yes.
at a population level, probably germline is better, but individual level.
But given the outcome that germline can fix the problem for future generations, but what is the guarantee that this gene won't cause any other issues? The method that you have used won't cause or won't introduce new problems. So the reliability of gene therapy is very low. Scientists have attempted this. Scientists, I think since 1990, they have been using gene therapy experimentally. They might have worked, I think, at least 2,000 times so far, as per the literature. But children were dying with the specific problems. Like when you perform gene therapy, the normal copy sometimes may get introduced somewhere else, resulting in cancer. Some children after gene therapy, they died with cancers. And some people after gene therapy, they developed multi-organ failure. So there seem to be problems in placing the gene in the right place. It is maybe disturbing the other areas in our genome. Okay, so for safety and ethical and technical reasons, it is not being tried. Germline therapy is not recommended, although it might cure the problem for coming generations as well. But for safety, ethical, and technical issues, so we have not optimized it. We don't know how to properly introduce yet. We don't have enough control, so we are not still using it. So somatic cell therapy is what we are trying now. Only cure the problem So not at the embryonic level. Few experiments have been done. I'll tell you in the next example. Few experiments have been done to introduce that in the germ cells and our embryonic stages, so very early on. So every cell will receive the modified version of the gene. But what is normally done is somatic cell therapy, limiting that effect, good or bad, to the particular individual. Okay. Okay. There are different ways. So these are different targets, somatic cells and germ cells. So in this image, if you see it, there are two ways that they perform gene therapy. Like one gene therapy method is called in vivo therapy, another one is called ex vivo therapy. So these were in the news. I think in last few years we are hearing this in the news. In vivo means like basically introducing the gene of interest. So what do they normally do is they would collect. They have to use carriers. You cannot simply drop the gene into a person's body. Let's say a defective gene, you are trying to cure hemophilia. You cannot just simply throw or inject the gene into the patient's body. The gene won't be accepted. So what do? They actually place that gene into viruses. Most commonly, we use viruses as vectors or carriers. So viruses are routinely used as carriers. So this is a therapeutic gene in the red color here. So this in a construct, they place it into virus. And when you inject into the patient's body, that will enter into the patient's body and then eventually carry the gene to the cells and place them into the nucleus of the cells. So viruses are known to do it. You don't have to train them. Viruses, when you get viral infection, they get into your cells. They get into your nucleus. We don't have to train it. So what scientists have thought, why can't we just use virus modified? They don't use normal infectious virus. They use non-infectious form of viruses. They take the virus, they genetically modified. So they take the key genes of them, place our gene of interest into them, like your hemophilia gene, factor 8 gene, factor 9 gene. They place into it and then drop it into the human body. They inject into the human body. When that is done, it is called in vivo therapy. It is called in vivo therapy. It is called in vivo therapy. But this is dangerous. You don't know whether this therapy will work or not. How many times you experimented it. You don't know how the given person would respond to it. So instead of trying this directly by injecting through viruses into the patient's body, instead of in vivo, instead of placing into the body, because here vivo means body, into the body, we do ex vivo. So we want to perform it outside the body. Outside the body. What do they do here? Do you see the difference between this image and this side of the image? So here we actually have humans and their cells are taken. Like stem cells can be removed. Stem cells are particular somatic cells can be removed. Maybe skin cells, maybe WBCs. They can remove it and then perform this step. So meaning add this therapeutic gene through the virus into these cells, which are being cultured, like lab meat concept I was telling you, we can take the tissue and culture them in the lab, right? Culturing multiplies the cells. So in the process, you add this virus carrying the foreign gene and see if this foreign gene enters into it, and if these tissues can stay happily, can stay healthy, can stay without dying. So these things can be done outside the body. That is called ex vivo. In vivo means inside the body, ex vivo means outside the body. So they can perform this outside the body. They can perform it outside the human body. It's called ex vivo. Which is more safer, in vivo or ex vivo?
Ex vivo.
Ex vivo, more safer, because we can see the problems here. Some more tests can also be done, whether it got integrated as it's supposed to. because not all cells may receive the gene in the same manner. Some may receive it and place it in the right place, some may not. So you can do those tests, and only those cells that carry gene as it's supposed to, they can be used. So we have option to analyze the cells before placing into human body here, but in vivo we don't have it. So the therapies that we do are ex vivo therapies, not in vivo therapies. Okay, right. So with that, but has it been done? Has it been in use? Did you hear about gene therapy being done anywhere?
Yes, sir. I think the one HIV person got cured or something. Not sure whether it's gene therapy or stem cell treatment.
Right. I think yes, it was very good. It was stem cell therapy where they used. They were giving treatment for HIV. They took stem cells from a person uh, who has a rare mutation showing resistance to HIV. We're going to talk about that next in designer baby controversy. Okay, we noticed that. So there are some mutations that can happen in some genes. Stem cells of that person have been taken. So this person who was cured for, I think it was cancer therapy, but his HIV problem was also cured. He didn't have to go on the medication again for HIV treatment. I think he was treated for cancer, but received HIV resistant cells. So mutations in their body, in the donor's body, so had HIV resistant gene. So that was introduced for cancer therapy. So he was cured for cancer and also for HIV.
Relapsed and died recently.
What is it?
The cancer relapsed.
Yes.
And he passed away.
Okay. So these are the problems that we cannot control. Correct? So, but experimentally, that's a great thing because nowadays we are finding those resistant genes. Maybe they can pre-try it or work it out how to use those specific stem cells to cure the problem. Okay, but otherwise, I'm not sure if you heard about this disease called SCID. Not last year, year before, maybe American doctors have cured 8 to 9 babies with this disease. Have you heard this disease? This disease has got a little complicated name. It is called SCID, Severe Combined Immunodeficiency Syndrome. Severe Combined Immunodeficiency. The problem is immunodeficiency. Is there a name on this? Yeah. So Severe Combined Immunodeficiency Syndrome. So don't worry about remembering the name or by hearting the name. Understand the problem here and how gene therapy and has been used. Why gene therapy is used? Because the therapy is new. Okay, even at that time, I think it was in 1990s, it was the first disease to be cured using gene therapy. Even today, even recently, they cured the same disease using gene therapy in children, but it's getting a bit more of use nowadays. In the previous cases, it was only experiments. So SCID or immunodeficiency problem. I'll briefly explain the story here. So this child has a rare genetic mutation in one of the genes. And that gene's enzyme, the gene codes for an enzyme, which is essential for the immune system to form and immune system to function. We have, I think we were briefly talking about it in immunogenetics, we have lymphocytes. Remember we talked about WBCs called lymphocytes, T lymphocytes, B lymphocytes. So a particular gene that has mutation here is responsible for creating functional lymphocytes, functional T lymphocytes to be more specific. So what happens if there is a mutation in that gene? Can this baby produce normal and working T lymphocytes? Because of the mutation, this child could not produce the working copies of the T cells. If no T cells, no cell-mediated immunity. So if your immune system is not well developed, what happens to you? As a baby, imagine that even when we have good and developing immune system, we suffer from many infections. Some children do die from infections. But if no such immune system has been developed, can we save babies? Can these babies survive? Question mark.
No.
No, they cannot survive because immune system is essential for us for our survival. A rich couple, you see in this, I think a Hollywood movie was made on this, I heard. Bubble boy syndrome, it is called. So this rich couple actually had a baby who died with this problem. And then second baby also got the same genetic problem. And you know what happened? What they have done? So they actually created a bubble for that baby. They created a sterile bubble for that baby that no infectious agent or a microorganism can enter into it. Baby was put into it, and in a sterile manner through these channels, they were giving food to baby. Baby was there for about 9 to 10 years in the bubble. That baby is called bubble boy. And that syndrome is called bubble boy syndrome on his name. Otherwise, it is KID, severe combined immunodeficiency syndrome. So unfortunately, what happened, this baby, I think one day doctor suggested to perform bone marrow transplantation on the baby, thinking that probably the T cells will develop after bone marrow transplantation. They took the baby out, baby died with an infection after 9 to 10 years. They saved the baby for about 10 years in the bubble and then died. So some diseases we have no cure. Then something like gene therapy must be attempted. And that is what had been done in this case. They attempted it, in later, I think Ashanti De Silva, there was a Sri Lankan girl, looks like. So this girl was treated for this, and then she got cured from SCID problem through gene therapy, successfully through gene therapy. But what happened in these children, I think, if I'm not wrong, her and also another baby in UK, USA and UK, two children died because of cancers or multiple organ failures. Therapy worked, they fixed the problem, but other problems got introduced into the baby in the process. Okay, this led to another set of issues. So like that, although theoretically this method works, technically this method works, but it results in other problems that we have no control on. So gene therapy has been on hold for a long time because of these issues. I think more recently, since last few years, you are hearing this in the news. Otherwise, experimentally, we hear rats eyesight or rats, vision issues or bone marrow issues. Specific diseases have been cured, but experimentally in the animals. For the humans wise, we didn't have too much of success. But more recently, I think I'll show you this in a document here. So in America, I don't think I've mentioned it here. Okay, I'll explain that to you. So in US, in National Cancer Institute, they have used a gene therapy, okay, to re-engineer immune system cells called lymphocytes. Okay, thereby curing cancer, successfully curing cancer for the first time using gene therapy has happened a few years ago in the USA. And also commercially, companies like Novartis, I'll explain this, and also what is called Kite Pharma. So two scenarios, Novartis and Kite Pharma. Novartis developed what is called Escarta. No, not Escarta, Kimraya. So Novartis developed what is called Kimraya, and Kite Pharma developed Escarta. So these are the methods. So same concept that you see here, re-engineering the immune system cells like lymphocytes. What they have done, they performed a gene therapy for cancer. How does gene therapy can be done for cancer? Now what they have done here, they took the immune system cells. They took the immune system cells. They took the immune system cells like T cells, and what they have done, they genetically modified it. They took the T cell, they genetically modified it. So genetic modification has been done in such a way that, so they introduce a new receptor, They introduced a new receptor. What did this new receptor done? So this new receptor, it's like a new molecule to find tumor cell. So I'll tell you what it had done. So they took the T cells in the laboratory, they genetically modified. And this genetically modified one was placed back into the patient. So the genetic modification resulted in a new receptor. Receptor is a binding molecule. It's like COVID-19 binds to a specific receptor, ACE2 receptor on our cells, on lung cells and other cells. So this is the interaction point. So what they have done, they created a receptor like that on the T cells. And what these T cells were doing, the T cells, when they were introduced into the patient, they are able to now go to cancerous cell and identify it and destroy it. Because T cells naturally have the ability to destroy the cancerous cells. Like I was explaining you yesterday, so that they have ability to release cytokines, chemicals, to destroy the cancerous cells and other infected cells. So, but the problem is, although we have that ability in our cells, they were failing to do so. So because of this gene therapy and because of introducing a new receptor on the T cells, now T cells are augmented. They made better at identifying the cancerous cells. So when these are placed into the patient's body, they were looking for cancerous cells and destroying them. So like that, they redesigned our own process. They augmented the ability of the T cells to detect the cancerous cells. And no side effects in this, because radiation, chemotherapy, you are exposed to a lot of radiation, exposed to a lot of chemicals, okay, which damages normal cells as well. Whereas in this particular case, this is called immunotherapy. It's called immunotherapy. The therapy resulted in the augmentation of existing T cells, thereby curing the cancer. Thereby curing the cancer. The gene therapy is now raising hope that cancers can be cured, but at the moment they are so expensive. I think they would cost, if I'm not wrong, at least 200,000, 300,000 US dollars. So not affordable at the moment. And experimentally, I think FDA approved them. They're getting into the market in the developed countries, but still long way. But Indian government is recently has come up with the guidelines to perform gene therapy in India as well. So they're making it legal and they're promoting research so in a safe manner. Such a research can be done in India. Okay, so gene therapy is happening. This is also a new way of treating problems. Of course, has been happening since 1990s, but now we have more success with it. Okay, so yes, there are many problems as well. I was telling you already, couple of problems, multi-organ failure, cancers, and our immune system may recognize the viruses and destroy them. So we don't, our system may not allow the viruses to actually introduce genes. So immune responses are the major problems. And sometimes, somatic cell therapy, when we do, only short-lived. So it may be good for a particular individual. So it may not solve completely the problem. And sometimes it's not one gene that gets problematic, maybe multiple genes involved in it. So modifying multiple genes at a given point of time is difficult. So multi-gene disorders cannot be handled using gene therapy. Like that, there are several limitations of gene therapy. Okay, right. So the last one here is the genome editing tools. We're going to learn about genome editing tools. I'd like to briefly explain you about the CRISPR-Cas9 system here. Genome editing tools and CRISPR-Cas9 as part of it. Because in gene therapy, what we have seen as a problem, that not being able to incorporate the gene where we want. So we are missing the target sometimes. We are dropping the gene somewhere else, resulting in cancers. So specificity is a big problem in gene therapy. But looks like we have now a new upcoming field called genome editing, may be of great use in gene therapy. Okay, as part of this, we will talk about CRISPR-Cas9 system. CRISPR-Cas9 system. What is CRISPR-Cas9 system? It's a type of genome editing tool. It's a type of genome editing tool where scientists have found, I think first I'll give you the abbreviation on this, CRISPR, although it sounds very crisp, but its abbreviation is not really crisp. So CRISPR stands for clustered, regularly interspaced, short palindromic repeats. Okay, too long a name to remember, right? But let's dissect it. Let's see what does it mean. So I'll ask you questions and you answer me, okay? So I don't feel sleepy and you don't feel sleepy. Right? Is that a good deal? What are palindromic repeats? Sunil, have you heard about palindromes?
Yes, sir. Like Nitin, from front or back, you get to get the same name. NITIN.
NITIN. I didn't hear about this word. Okay, good. Nitin reads the same. Okay. Malayalam. Does it read the same in both the directions? So palindromic repeats, palindromes are those that read the same in both the directions. Malayalam, Malayalam. I basically, Nitin, you're right. Malayalam. So they are examples of palindrome repeats. Palindromes. Repeats means they repeat. Same sequence may repeat multiple times. Like I'll give you an example of a repeat. GAA. Okay. GAA or GATTAG. So read it in both the directions. GATTAG in this direction. Other direction also same. GATTAG. Such sequences are found in DNA of bacteria, because these CRISPR sequences were found in bacteria. They were found in bacteria, and where they found out that GATTAG like sequences, like this palindrome sequences are found, and they are found in repeats, not one sequence, but it was repeating multiple times. Like this. But what do you see? So they are short. Short in the sense only few letters. Of course, we have taken the short ones, maybe a few letters, 8 letters, 10 letters or so. We have taken about 6 of them. That is good enough. So they're short palindromic repeats, small regions, okay, small letters, few letters, sorry, and palindromic in nature. So they repeat like that. But they are not present next to each other. They are regularly interspaced. Interspaced means there is a gap, same amount of gap, like this. So there's a gap between them. Okay, so they're short palindromic repeats which are regularly interspaced and they are clustered. Clustered means they are found in groups. Like this in bacteria, if you see, they're found in several groups. A package of this is maybe present here in the bacteria, somewhere else in the bacteria, like they are present in clusters. Such sequences are called CRISPR sequences.
Is the gap filled by some other letters?
I'm going to come to that, yes.
Or
Yes.
is there a gap?
No, there is no gap. Otherwise, how does DNA stay like that, right? So there are some other sequences in between which are known as spacer sequences. So the gaps are filled with other sequences, spacer sequences they are called. And where are these sequences coming from? Interestingly, the spacer sequences are viral sequences. So we are talking about bacterial DNA, and between these palindromic repeats, there are sequences that belong to viruses are present. What are they doing there? How did they get there in the first place? Shweta, do you know anything about it? Did you place those viral sequences there? No? Okay, it's not you. Sunil, it's you. Vishal is hiding. Maybe Vishal. Right, Vishal?
It's viruses themselves, I think.
What is it? Sorry.
I think the viruses themselves must have gone into that bacteria.
Right.
Some
What type
point.
of virus is going to bacteria? Do you know the specific type of viruses that get into bacteria? You do hear this in the news. These days they're talking about, instead of, because antimicrobial resistance is a growing problem for antibiotics, why can't we use viruses that can infect bacteria? They're saying if you get a bacterial infection, take viruses which attack bacteria. What type of bacteria are those? Alien-like bacteria. Have you ever seen them? Bacteriophages.
Yes, sir, bacteriophages.
Bacteriophage. Bacteriophage are the viruses which infect bacteria. Those bacteriophages sequences got in between these sequences. This is Nobel Prize winning research. This year's Nobel Prize. CRISPR-Cas9 system, and a hot topic for UPSC. Came at least twice in UPSC prelims. Very, very important topic. And this is something that is causing more ethical debate now on biotechnology and genetic engineering. Okay, so bacteriophage sequence got into these palindromic repeats. And the story is this. Let me now put the things together, pieces together, and explain you the puzzle. Fix the puzzle here. Okay. So what happens? The scientists found out that bacteria is commonly attacked by bacteriophage, and when bacteriophage attacks the bacteria, it drops a viral sequence. It drops the viral DNA into it. It injects the viral DNA. It injects its software into the bacteria. Okay, it injects the viral DNA into it. Let me see if I have an image to show you here. Oh yeah. So this is how bacteriophage looks like. Okay, so this is bacteria. What happens? Bacteriophage normally injects its DNA into the virus. So it's like this. I think this is what you are seeing here. We should begin here. Bacteriophage, its DNA got introduced or injected into bacteria. So what happens? This sequence is stored here. The black sequences are palindromic repeats. The colored ones, each colored ones belongs to one particular type of virus. So green one, orange or reddish one, purple ones, blue ones, like that. So as soon as the viral DNA enters into a body, a piece of this is kept within the bacterial sequence. What is the use of keeping it? It's like a memory. Why? Because every time a virus again enters into the bacterial body, what will it do? It will transcribe this part. Let's say green part, a green DNA virus is entering. So this portion gets copied onto RNA. An RNA will be made. This is called a CRISPR RNA. So CRISPR is an RNA component usually copied from the genetic component present in bacteria. So this part belongs to whom? Virus, right? So CRISPR containing this viral DNA component is formed, and it goes and finds the viral DNA. So whenever viral DNA is introduced for the second time, this RNA will go and match it. It's like a probe that we were talking about the probes yesterday as well, right? So like probe, it will go and bind and identify the viral DNA when it enters for the second time. But along with it, it will take its best friend named Cas9. So it will take a best friend called Cas9. What is the role of Cas9? Cas9 is like a chopper. It's like scissors. As soon as this guide RNA will bind it and says it is viral DNA, it will go and chop it. So it'll eat it, it'll bite it. It's like a good friend in the childhood. If someone hits you, they'll go and bite them, right? So it's like that. Cas9 is like a enzyme which goes and cuts the viral DNA. So every time the second viral DNA repeatedly enters into the bacteria, it is protected by this CRISPR system. So CRISPR is like an immunity for bacteria against bacteriophage. So how is it working? It has palindromic sequences between which it saved viral pieces as soon as virus enters into it, and somehow it survives from the infection for the first time. Second time when the same virus comes, it's like how we take vaccines. Okay, we produce that this bacteria produces this CRISPR RNA, which will go and find the viral DNA, and along with it, Cas9 will go and cut it. So like that, viral DNA will be cut, chopped, and killed. So like that, it's a protection system that is found in bacteria. Such a system is called CRISPR-Cas9. Is it really crisp? It is not, right? But are you able to follow the concept here? Any difficulty in understanding what I explained you so far?
So is it viral DNA or RNA that gets embedded into?
Gets embedded. So this is a DNA virus. Bacteriophage is a DNA virus. The DNA part gets incorporated into DNA. So DNA gets incorporated, but DNA produces RNA. So the first part here is DNA. This is DNA. This is RNA.
So,
Hmm.
we have learned about the restriction enzyme. They also work like scissors. So Cas9 and restriction enzymes are similar.
No, they are both are scissors, but different types of scissors. So restriction enzymes cut at specific sites, whereas Cas9 cuts wherever CRISPR tells it to cut. So they both can cut, both are nucleases, so DNA, RNA cutting enzymes, but work differently. So here they require CRISPR, they are there, and they have a particular sequence found in the DNA. Wherever the DNA sequence is, it cuts it. Here it cannot cut itself, it requires CRISPR to tell it where to cut it. But otherwise both are, we don't call them as restriction enzymes, we call them nucleases. Those ones are restriction enzymes that we learned in the last class. Okay, but similar function, as you notice.
So can you repeat once more? I didn't get it completely.
Sure. Okay. Actually, I'm going to send you a video on this. Okay, there is a video made by scientists at MIT on this. A great video, excellently made, and along with some basics that they cover, and they show great animation on this. How does virus sits on it? How does it inject it? Where does it go? Enters into the DNA. What happens then in the DNA region? Except they give a little bit more details. So ignore those details. They talk about not one component of RNA, two RNAs come together, but imagine it's a one RNA for our purpose. And they talk about more than spacer sequences. They talk about PAM sequences. Ignore them. The idea is this. So get the idea of what we discussed, get it as a visual. I'll paste it as soon as I post it in the group as soon as I'm done with the class. So you watch it. But I'll give a review on this and then also watch that video. That might help you understand it. So what are we talking about here? So bacteriophages are known to infect bacteria. First point. Second point, in the process, bacteriophages insert or inject their DNA into bacteria. Okay, what happens when viral DNA enters into bacteria? Of course, viruses supposed to kill bacteria. Their job is to infect bacteria, kill them, just the way COVID-19 is planning to kill us or has been killing us. Same way, bacteriophage should kill the bacteria. But what normally happens here, so when viral DNA enters, bacteria somehow has to survive. Otherwise, what happens? All the bacteria would have been vanished by now. But that's not the case. They're surviving. How are they surviving? One method that scientists found out that whenever new viral DNA gets into bacteria, a portion of the viral DNA is stored between what is called palindromic sequences. So like that, this particular sequence showing the presence of several viral DNA sequences. So bacteria remembers those sequences by placing the particular DNA portion between the palindromic sequences. Okay, right. So here these black ones are palindromic regions, these colored ones are spacer sequences. So spacer sequences are the viral sequences, viral DNA sequences. So what happens, let's say a bacteriophage started affecting it again. Second time infection has happened, a bacteriophage has introduced their DNA into it, but bacteria are now already prepared towards it. What did it do? So it actually transcribed this region. If a green colored bacteriophage is coming here, so this green region was used to make an RNA called CRISPR RNA. So it transcribed this region, forming CRISPR RNA. And this CRISPR RNA, along with its friend called Cas9, went to the viral DNA and matched it. So CRISPR RNA went there and said, okay, now I found the viral DNA. Cas9, it's your job. Cas9 will chop that wherever this viral, wherever CRISPR RNA bound, that region is basically damaged by or broken by Cas9. So together, CRISPR-Cas9 system kills or destroys the viral DNA, thereby protecting bacteria. So it is like an immune mechanism in bacteria. So CRISPR RNA guides the molecular Cas9 machinery to target this. So this is what you see here. This is a CRISPR system. This is viral DNA system. So CRISPR system matched viral DNA, Cas9 cuts it. So thereby destroying the viral DNA and no infection will happen. So that's how they get protected. Is a concept clear? Could you follow this, Sunil? Kind of?
Yes, sir.
Good. So the video should help you further, okay? I'm going to send you that video link. That should help you further. So what else? Yes, now the question is, why are we learning about bacterial system? And we are calling it genome editing tool. So now this is the idea. Now I'll tell you how are we using it? What is the use to us? So we are learning this concept from bacteria. Scientists who received Nobel Prize last year, okay, 2020, there is Nobel Prize in October and November time that they received, was in chemistry. It is given to CRISPR-Cas9 system. What did they find out here? They found out that we can mimic the process in process that is happening in bacteria. Like you can take the concept of CRISPR RNA, but instead of targeting viral DNA, can we target the region that you want to modify? Let's say hemophilia is happening because of factor VIII gene. Can we go and specifically modify that region? So they are working on, they designed tools, CRISPR tools, in such a way that just the inspiration is coming from bacteria. But what they are doing, they created a guide RNA. This guide RNA will now target something that you want to perform gene therapy. It's a the factor VIII gene. So like that, you can design sequences where guide RNA will match to the region that you want. Okay. So scientists create a genetic sequence called a guide RNA that matches the piece of DNA they want to modify. Second, this sequence is added to a cell along with the protein called Cas9. Both this guide RNA sequence as well as the Cas9, they place that into human cells. They place that into human cells. Okay. So what do they do? Cas9, CRISPR will go and bind to wherever factor VIII is, for example. And Cas9 will go and chop it there. Right. So exactly what they did in the bacteria, chopping the viral DNA. But here you are actually cutting human DNA, human target DNA. Then what happens? So two outcomes are possible. One, whenever our DNA is cut, we have a repair system in our body. Whenever DNA damage happens in our body, repair system kicks in. It's like a rescue system. Service centers, for example. Your car broke down. You call the service center, they would send you people, mechanic to fix it. Similarly, we have mechanics called enzymes, DNA repair enzymes. They'll come to the picture, they'll fix the problem and go. In the process of fixing, two things happen. One, let's say we have normal version of the gene, but because of cutting, this repair system will add mutations. They cause problem in the gene. Because normally they say this is error prone system. When they try to fix, add nucleotides, they make mistakes. Is it good or bad? Making mistakes is bad. But what if you want to create a model organism where genetic sequence has to be modified? You want to, we call it knockout system we call. Okay. So what happens here? You actually modify the gene in such a way that mutations are introduced. So this is good if you want to create mutations and make that gene non-functional. But this is not what we want in fixing the genetic problem. So what do we want in fixing genetic problem? We want the mutation to be fixed. So scientists say that, so along with the CRISPR RNA and Cas9, introduce a normal copy of the gene. So basically perform gene therapy, but along with gene therapy, you introduce the CRISPR system and CRISPR-Cas9 system. So what happens? CRISPR cuts it, CRISPR identifies it, Cas9 cuts it, and then when repair system comes into the picture, they may take the normal copy of the gene which is in the vicinity, assuming that it is the version of the DNA that it has to fix. So it may switch. I know it is very difficult to imagine, but I'll repeat this. So it may use the normal and functional copy of the gene that you have introduced as a template and fix the problem. So there's a small percentage of chance, but it was observed. We don't know how exactly it happens, but this is how scientists explain it, but it has happened. So where scientists added CRISPR system, Cas9 system, and a new gene, and the new gene template, a new gene is used as a template, and the repair system fixed the problem by changing the mutation. And why did they come? They came because Cas9 cut it. In the process, they fix the problem. It may happen in 1 out of 100 cells, but we don't mind. You perform this on 100 cells at a given point of time. Look for those cells that actually created the expected change, and take those cells and place them into the patient. So patients will be happy because now his cells are modified and the genetic mutation has been reverted. So that is how scientists are nowadays using CRISPR-Cas9 system. It is becoming so popular. MIT said this is the most and I think influential or famous technique developed in biotechnology for this century. 2012, I think, when they discovered this, they actually they discovered it before that, but 2012 they got with the idea of modifying our genomes, human genome, plant genome, microbial genome. They came with this idea. So did you understand the concept here? I'm repeating this. So we are following or mimicking what happens in bacteria. So scientists can create CRISPR RNA, but it will match the target gene, maybe human gene, plant gene, bacterial gene, whichever you want to modify. So CRISPR provides that specificity. CRISPR guides the Cas9 to the specific sequence. And what does Cas9 do? Cas9 will go there and will chop it. In humans, for example, the chopped DNA will not stay like that. In bacteria, phage DNA will die. But in humans, we have a repair system that comes in there to fix the problem. But in the process, one, they can create mutations, or if you provide normal copy of the gene, they may take the normal copy of the gene as a template and fix the problem. So wherever the mutation is, but mutation is not present in the normal copy of the gene, so it will look into the normal version of the gene and then fix it. So this is what the hope, and experimentally scientists have proved that it works. But how they explained it is this. This is a bit complicated to understand. Okay. So scientists did they really use it? Indeed, they did. In August 2017, scientists in USA, Oregon Science Health University, they've used CRISPR-Cas9 system to modify an embryo which carried a rare mutation. Don't worry about the name. Name is a bit complex here. They said inherited hypertrophic cardiomyopathy, some rare genetic problem that they have fixed using CRISPR-Cas9 system. So scientists have proved in USA that it can work. This is one scenario. Otherwise, forget about humans, but plants, microorganisms, CRISPR-Cas9 has become a common tool. They are modifying them like anything with CRISPR system. Very cheap, not very, very cheap, but economic, accurate. They're using it. But the problem appeared, ethical problem. The problem appeared when Chinese scientists have used it to create first human designer babies. So in USA, when embryos are modified to remove the genetic mutations, it is all right, it is acceptable. But for many, it is not digestible when they heard that Chinese have created human designer babies using CRISPR-Cas9 system. I'll take another 5 minutes, I'll wind it up. Have you heard about this story? Designer baby controversy, have you heard about? Okay, so what have they done here? Let me briefly explain this. Okay, so in this case, in China, I think a couple and the father is HIV positive. Here the story, I'm going to share this with you. Father is HIV positive, and they were worried that their children would also get HIV from the father. They approached this scientist, and what scientists suggested, scientists said with CRISPR-Cas9 system, we can create a mutation in the embryo. They said, go for IVF, in vitro fertilization technology. In the process of IVF, when embryos are made, so the fertilization happens in the laboratory, embryo is made. They said we can make changes to embryo using CRISPR-Cas9 system, which is a very accurate tool. So what they have done, they actually modified baby embryo has been modified, targeting what? There is a particular gene called CCR5 gene. So this is a gene, I think Vishal, you were asking about someone got cured for HIV. So in this case, that person had CCR5, mutation in CCR5, because CCR5 is an essential gene for HIV to enter into our body. Because CCR5 protein, what the CCR5 protein is the protein that interacts with HIV. When HIV makes interaction, it introduces its RNA into our cells. So for its infection, we need CCR5, functioning CCR5. So scientists thought, why can't we just modify this? Chinese scientist, his name is He Jiankui. His name is He. Okay, He Jiankui. So he modified CCR5 because already it is known that CCR5, if you trouble the CCR5, modify the CCR5, resistance will be created. So he said, why can't we use CRISPR system? So Cas9 RNA will target CCR5 and Cas9 will cut the CCR5. CRISPR system will identify the CCR5. So like that, they targeted CCR5 gene. So they took the embryo, IVF they performed, took the embryo, added CRISPR system to target CCR5 gene. So specifically CCR5 gene is modified. And that embryo and later has been placed into that biological mother or surrogate mother. It resulted in twin babies, twin girls. Nana and Lulu are the names of these twin girls. Nana and Lulu. These twin girls formed out of this experiment. And they are resistant to, or at least they are supposed to be resistant to HIV. So scientists have created first human designer babies who are resistant to HIV using CRISPR-Cas9 technology. Good news or bad news? There's a Hindi movie, right? Good News, where they switch in the IVF, they switch the donors, I guess, donor samples. So this is where ethical dilemma comes into the picture. Yes, I think it is all right to modify something that can cause problem, but not for HIV, because HIV we have other treatment options. Antiretroviral therapy we have viral drugs to not cure HIV but manage HIV. But for children, if mother is known to be HIV positive, you can start treating mother and also child as soon as child is born. And it has been shown that successfully you can prevent HIV entering into baby's body. So HIV transmission to the babies, this is called vertical transmission, can be prevented. So why take the risk of this when there is no guarantee that this method will definitely work? I was telling you this is accurate, more accurate, but it is not 100% accurate. So this method is not 100% accurate. So scientists, I think Nobel Institute, do you know what is Nobel Institute? In Sweden, Stockholm, Karolinska, where Nobel Prizes are given. In this Swedish institute, scientists have shown that when CRISPR-Cas9 is used to modify animal body, animal tissues, lot of mutations, additional mutations got introduced. Lot of DNA damage has been noticed. So there is no guarantee that this method will accurately work. It has ability to work, but we have not figured it out yet. So it is too early to use it on humans. So in haste, in urgency, Chinese scientists have tried to create human babies, designer babies. When other studies are pointing out that we cannot be 100% right with this technology, then now who will take care of that baby? If something happens to those babies, who will be responsible for it? Parents or scientists or regulatory bodies who allowed them or who ignored them when they're doing research. Now, what did Chinese government did? Chinese government said we never allowed them, we never permitted them. They actually canceled their licenses because both of them are doctors. A team is doctors. Those couple of doctors' licenses got cancelled. They put them in the jail and fine. But what about the babies? Is it ethically correct? You are interfering, doesn't matter. Gene therapy, cloning, IVF. Are you not interfering the natural process? For some people, it is not acceptable. Why are you interfering without knowing the implications of what you are doing? The CCR5 gene may have other functions. You are disturbing it. Just because you found one switch, but the switch may lighten many and may have many connections. May be useful for a fan and a light. You are trying to switch off the light, but also you switch off the fan. So you don't know, because sometimes some genes can play multiple roles in the body. Okay, so without knowing the long-term complications, trying on humans is not acceptable. Maybe trying in the embryos to prove it is all right. Maybe trying on something like SCID, where the child is going to die, is all right. But here, these kids have future, and these scientists have ignored, maybe for the sake of fame. They were never discussed this with scientists. They never published this work for verification. They went to a conference and discussed this in a conference. That's it. And which is not acceptable to many people, including scientists who invented this technique, who used this technique for gene modification. Jennifer Doudna and Emmanuel Charpentier, who received Nobel Prize. Read CRISPR-Cas9 Nobel Prize story on Hindu or even nobelprize.org. You will read about those stories. So those people are also very, very unhappy that scientists already used it for creating human designer babies. Okay, it is too early. Maybe for future, we are not against that concept, but it is causing a lot of ethical issues. So these are some of the issues that biotechnology can cause. Biotechnology can be used and misused by people. In this particular case, they created designer babies without knowing what will happen to them through this method. And cloning, we also cloning animals. What if someone wants to clone humans? Maybe someone wants to clone Osama bin Laden. Maybe cloning Abdul Kalam is acceptable, cloning whoever you want. Okay. But maybe some like cloning Osama bin Laden or someone who can create more problem to us. Is it acceptable? First of all, leave out whether you can create Osama bin Laden or Abdul Kalam. Maybe not practically possible, because you may create a copies of them, clones of them, but they may not become Abdul Kalam. You may create a copy of Abdul Kalam, but he may not behave like Abdul Kalam, may not have intelligence like Abdul Kalam. That may not happen, because their nature and nurture debate. Nature cannot define everything. It has to be nurture as well. So you may not be able to create Abdul Kalam with that. But in those type of methods can be misused by other people. It's like dynamite. Dynamite was created, Alfred Nobel created it. What happened? It was used for destruction. So like that, there are ethical issues associated with the biotechnology and genetic engineering. Be aware about them. This year they asked about it. Take few examples, designer baby controversy. In case they have done, the long-term complications are not known. So it is ethically unacceptable. And we are cloning humans, but this cloning humans can be misused for cloning, some other organisms who can actually cause more destruction to the society. You are interfering in the natural process of creation without knowing further complications. Not acceptable to many. GM crops, transgenic animals. GM crops reducing biodiversity. We are actually harming the diversity of nature because people prefer genetically modified crops. Not acceptable. We are killing mosquitoes. We are actually not killing mosquitoes. We are creating genetically modified mosquitoes. And they are sterile mosquitoes. In Brazil, in Australia, in UK, they introduced this. And these mosquitoes are sterile. So when they went into the population, they reduced the mosquito population. Scientists went further on creating sterile female mosquitoes. What do? After a couple of generations, whole mosquito population will be wiped off, creating ecological balance. Is it ethical? So many things that they are doing in biotechnology raising these ethical issues. Be aware about some of them and be able to answer it in case if you get this in the exam. Okay. I guess. So this is what I want to talk to you today. We discussed a few different technologies here. We talked about stem cells, gene therapy, and CRISPR-Cas9 technology we discussed about. These are examples of things. And before that, I talked to you about RT-PCR. Okay, better diagnostic, better treatments, novel ways of treating problems, genetic problems, cancers, infections are all possible using DNA technology. Okay, a brief summary that I'm going to provide you along with this information to you. And I will also post you right away the video that I wanted to watch related to the CRISPR-Cas9 technology. Okay, any questions, guys, before we close the session for today? So these topics we discussed today are also important for the prelims in science and technology developments and also for GS3 paper 3. Okay, right.
So,
Yes.
did you get a chance to look at the answer scripts?
No, not yet. So tomorrow evening I'm going to look at them. So on the weekend I'll share the information with you. I'll first give you the personal feedback on them. Okay, so I will definitely do that. They are on my list. So tomorrow evening, after my class, I'm planning to do that. Okay, and I'll post individual information will be posted to you. Sir, actually returning them on the website, on the course page, or they're sending personally?
Sharing a Google Drive with all of our transcriptions in one place so that we can refer to others' copies as well. And
Okay, so Google
understand.
Drive folder will be created and shared with you. So meaning I need your email IDs to permit you to see it. Am I right?
I think he's sharing a link with.
Yeah, I think he made it public.
Whoever have the link can access it. Got it. Sure, I can do that. Or else I think I also have your email IDs on the course page. Okay, sure. I think it's easier to create the link. I'll do that. Yes.
Or even Telegram should be fine. So even if you can share it
Yeah,
on
Telegram.
is also
Okay, all of yours. Oh, in the course page, in the course Telegram group.
fine.
Okay, so all of theirs, all of yours together. Got it. Yeah, it doesn't matter, both of them.
But sir, a link would be better because we can refer it afterwards as well.
Same thing with Telegram, right? You can refer it afterwards.
I happen to miss out on some of it. Okay, fine.
Yes, I'll do Google Drive. No issue. I can do it on Google Drive. Right, guys. So tomorrow we're going to begin with 9.6 genetic markers. Okay, so I'll give this to you today. And the next week, I think we were talking about test, right? Did I tell you the date? Any day?
I thought of Thursday.
Friday.
Thursday or Friday? I think I must have written it. Anthropology next Thursday, 9.2, 9.4. Okay, so weekend I will give you the papers so you'll know how to write. Meanwhile, start preparing for the 9.2, 9.4 as well. Okay, right, guys. So I'll share the handout for the DNA part today and link right away. Okay, right. Thank you.
Thank you, sir.
Thank you.
You're welcome.